Hello everyone, my name is Shane Roesemann. I'm a graduate student in the lab of Jason Kwan at the University of Wisconsin-Madison. Today I'll be talking about taxonomy-aware marker gene sets improve metagenomic binning through accurate purity and completeness calculation. So, metagenomic sequencing is useful for profiling unculturable microbes and seeing interesting things that they're able to do, like make novel drugs. However, to make these interesting conclusions, we need accurate binning. So, through the process of sequencing, the genomes of the microbes get chopped up into little bits. They're then assembled into contigs, and then to rearrange these contigs into bins that approximate the original genomes is the step of analysis that we're looking at, this binning step. So, Automata 2 is a binning algorithm that uses this iterative clustering design where bin purity and completeness is calculated with each iteration. So if you look at the bottom left here, I have this Automata 2 workflow. It talks about the entire workflow of the program, but I like to focus on this genome binning step where contigs are clustered together using a clustering algorithm, and with each iteration, the clusters are considered putative bins, and they have their purity and completeness calculated. The way that we calculate it currently is to use a static set of 139 bacterial single count marker genes. And if the bin passes these purity and completeness threshold, they're then considered usable for downstream analysis, and unclustered contigs go through subsequent iterations of clustering and calculation. Now, there are better ways of calculating purity and completeness. CheckM is one way, is a program that uses a much more expansive set of marker gene sets. So they have marker gene sets that are taxonomy aware, and so there's a different set for each node of the bacterial taxonomic tree. So we would just slot in CheckM into this step of Automata, but we fear that the iterative nature of our clustering would make the runtime of Automata to be too slow. So we're going to try and generate our own taxonomy aware marker gene sets to improve Automata 2's ability to bin by changing the way that we calculate purity and completeness. So the way that I'm doing that is I'm taking 62,000 genomes from GTDB, which is a curated database of genomes and assembled genomes that represent the taxonomic breadth of the bacterial tree. I'm annotating them with PFAM and identifying single count marker genes. Now, for each node of the bacterial taxonomic tree, I'm finding single count marker genes that are found in 97% of downstream genomes. And if it passes that consideration, then it is now part of the marker gene sets for that node. So for each node of the bacterial taxonomic tree, I have a new set of marker genes that I'll consider. Now, luckily, Automata 2 has this taxonomic information as part of the binning algorithm. So as it goes through this iterative step, the taxonomic ID is estimated, and then the corresponding marker gene set can be used to calculate completeness and purity. I will be considering two data sets to evaluate these new marker gene sets. I have a 78 megabase pair set, which is a simulated data set of genomes taken from NCBI that are chopped up to represent kind of contigs. And then I have this Mix51, which is a synthetic community taken from a library of isolates that were grown in a lab and sequenced together. And it includes some closely related strains, which is very confusing for these types of taxonomy-aware algorithms. Now, for the 78 megabase per data set, I'm seeing really comparable binning metrics, such as completeness and purity, but I also have some metrics describing the size of the contigs that come out, in addition to the precision and recall. The precision recall I'm able to uh, calculate because we know exactly the genomes that are going into these binning algorithms, and so the bins that come out should represent the genomes that went in. And I'm seeing some really similar results between the old static 139 gene set and my new dynamic marker gene sets with this 78 megabase pair data set, which is really encouraging for me because at least I know that my new totally different way of calculating purity and completeness will not totally ruin the way that Automata 2 is able to bin contigs. With the Mix51 data set, I'm seeing really, really similar results between the static and dynamic methodologies. They're nearly identical, and this is even more promising because I feel that the Mix51, with some of its really confusing isolates, are kind of more representative of an actual metagenome. So I would like to recognize that this is a work in progress, and ideally we would see an increase of the ability to bin contigs, of course. The next steps are to refine these marker gene sets to improve the binning outcomes. The first step that I'm going to look at is genes that are consistently close to each other in these genomes. And this method, looking at this idea of co-occurrence. But I will also expand it to consider multi-count genes, and I have some ideas of some test data sets to further evaluate Automata 2's new binning methodology. And with that, I am finished, and I'd like to thank you for your time.